Module B, Key Concepts. As you remember, we went over several trial examples in Module A, and we familiarized ourselves with common multiplicity problems that arise in clinical drug development. So let us now put a more formal structure around this. There will be 3 sections included in this module, as you see on the slide. Section B1, Inferential Goals. The reason we're going to talk about inferential goals is because the choice of statistical methods used in multiplicity problems is driven by the clinical objectives in each particular setting. It depends on the underlying inferential goal. For example, multiple analysis may be treated as independent entities, or they could be treated as components of a single overall analysis. In this section, we will define 3 different inferential goals, and we will talk about appropriate multiplicity adjustment procedures. This includes at least 1 procedures, and we will also call them multiple testing procedures, all or none procedures, and finally global procedures. Section B2, error rate definitions for multiple testing procedures. The most relevant inferential goal I would like to point out in confirmatory clinical trials is the goal based on at least 1 testing, and an important feature of all at least 1 procedures is that they lead to error rate inflation, and therefore they require multiplicity adjustment. To choose an appropriate adjustment, it is important to select the correct, most meaningful error rate definition, and this is exactly what we're going to discuss in this section. And finally, section B3, selection of multiple testing procedures. In this section, we will be ready to tackle the most important problem, really the billion-dollar problem. We will go over criteria for selecting the most appropriate multiple testing procedures for a particular at least 1 testing problem, and we will define a classification scheme, actually several classification schemes, for at least 1 testing procedures, or as we will call them from this point on, multiple testing procedures. So let us take a look at each individual section included within this module. Section B1, Inferential Goals. Within this section, we will discuss inferential goals in trials with multiple objectives, and it is important to build a formal classification scheme here because inferences used in a particular multiplicity problem depend on the underlying differential goal, as I explained a minute ago, and we do not want to get the problem misclassified and choose the wrong tools for addressing the multiplicity we're facing. And unfortunately, it happens more frequently than you may think that multiplicity adjustment strategy chosen in a particular trial may not actually match the actual multiplicity problem, which is going to lead to very inefficient analysis. The trial sponsor can consider 3 different sets of inferential goals in a clinical trial. The first one is the individual analysis separately lead to a successful outcome. This is what we normally think of when somebody says a multiplicity problem. And more formally, it is known as an at least 1 multiplicity problem, and within this setting, the sponsor needs to use at least 1 procedures, or as we said, simply multiple testing procedures. Secondly, individual analyses may jointly lead to a successful outcome. This would be known as an all or none problem, which means that all tests must be significant simultaneously, or the trial sponsor will be out of luck. All or none procedures need to be used in trials with this inferential goal, and the final type of an inferential goal occurs when the overall analysis of multiple objectives may lead to a successful outcome. This is known as the global testing approach, and it calls for global testing procedures. Let us begin with at least one multiplicity problems. As this simple diagram shows, we have multiple analyses or multiple objectives that are represented here by two orange boxes, and each analysis is independently clinically relevant. This means that each endpoint test or each dose control comparison, each population test independently provides proof of efficacy. And by independently, I mean here independently of each other. For example, a novel treatment may be highly effective at one dose, and there may be no signal, no evidence of a positive effect at another dose. But this will not damage, so to speak, the positive signal at that first dose, because each analysis is an independent entity within this inferential framework. And it's important for us to remember that the trial's outcome is declared positive if at least one analysis produces a significant result. What we saw on slide 6 was a simple diagram, and more formally, this problem is known as the union intersection problem. Why union? The objective of a clinical trial in this at least one setting is to reject the global hypothesis of no treatment effect in favor of a union of alternative hypotheses of a beneficial effect. So the key word here is union, and this is an example of a classical multiplicity problem, and we will focus actually on at least one procedures or multiple testing procedures in this online training course. The reason this class of multiplicity problems is so important to us is that at least one testing leads to an inflation of an appropriately defined error rate. The error rate gets out of control, it gets inflated, and thus a multiplicity adjustment is required. And I will mention quickly that a multiplicity adjustment is essentially a multiplicity penalty. It adjusts the significance levels for the individual tests downward to help control the error rate at the desirable level. And we will talk more about this class of problems in modules C, D, and E. And as far as examples are concerned, there are three quick examples I would like to use here. They were introduced in module A, and they help illustrate this particular inferential goal. Remember, we said that they were all conceptually similar to each other. Those three examples that, once again, we defined in module A. The first one based on the prostate cancer trial, the second one on the type 2 diabetes trial, and finally, example 5 based on the non-small cell lung cancer trial. These examples share the same inferential goal, and we will use multiple testing procedures in settings of this kind. This is a quick illustration of at least one multiplicity problems, or more formally, union intersection problems, based on example 1, based on the prostate cancer trial. And the delta 1 and delta 2 used in the slide are defined at the bottom of the slide. They are defined in terms of the true treatment differences based on the first endpoint, which was the overall survival, and the second endpoint in this example, which was progression-free survival. The treatment differences are most commonly defined using the logs of the corresponding hazard ratios, and delta 1 for overall survival is plotted on the horizontal axis, and delta 2 for progression-free survival is plotted on the vertical axis. The shaded area is the region in the parameter space which corresponds to the global null hypothesis. Within this shaded area, both treatment differences, both delta 1 and delta 2 are negative, and that means that there is no beneficial effect compared to placebo. The sponsor's objectives is to break out of this shaded area and to demonstrate a positive effect on one or both endpoints, which will be the global alternative in this particular setting. We are done with the discussion of at least one testing, and this slide discusses the second inferential goal, all or none testing. This goal is based on a stringent requirement that all analyses must show benefit, which means that all tests must be simultaneously significant, and only after that the sponsor can claim that the primary objective is met, which means that the overall trial outcome is a success. The all or none testing problem is also known as the intersection union problem. It is intersection union because everything here is a mirror image of the at least one multiplicity problem, which was known, as we said, as the union intersection problem. In particular, the trial sponsor needs to reject the global null hypothesis of no drug effect in favor of an intersection of alternative hypotheses of a positive beneficial effect. Once again, the key word here is intersection, and that explains why this particular class of multiplicity problems is known formally as the class of intersection union problems. And if we need a multiplicity adjustment in the intersection union setting to protect the error rate from inflation, as it turns out, there is actually no error rate inflation, and thus there is no need for a multiplicity adjustment. Going back to the trial examples that were introduced in Module A, you may remember that we saw the requirement to demonstrate a beneficial effect for all analyses, all endpoints in example 2, which was based on the Alzheimer's disease trial. In this particular case, the primary analysis involves two co-primary endpoints. And as we said in Module A, the FDA guidance defines co-primary endpoints as follows. It is stated in the draft guidance that multiple primary endpoints in the trial are termed co-primary when it is necessary to demonstrate a beneficial effect on each of the endpoints to conclude that the experimental treatment is effective. In this trial, the sponsor needs to establish a significant effect on both co-primary endpoints simultaneously, which means on the cognition endpoint and on the global clinical assessment endpoint as well. This is how the clinical win criterion is defined in example 2. The sponsor can claim significance on all endpoints, or the sponsor gets nothing. It will be a failed trial if there's only one non-significant endpoint test. The diagram on the slide shows the global null and the global alternative hypotheses in the Alzheimer's disease trial. In this example, the delta 1 and delta 2 again denote the true treatment differences for the cognition and global clinical assessment endpoints. As before, delta 1 is plotted on the horizontal axis and delta 2 is shown on the vertical axis. The shaded area again defines the global null hypothesis, and now it is very easy to see that it's much larger than in the prostate cancer trial. The global null hypothesis in this setting occupies 3/4 of the parameter space, and this is because within the shaded area at least one delta is negative, or sometimes both deltas are negative, and this is a bad sign for us in the context of the intersection union multiplicity problem. The global alternative hypothesis is defined by the requirement that the true effect on both endpoints must be positive, must be beneficial, and the parameter space for the global alternative hypothesis is quite small. It's only one quadrant, as you can see on the slide. Claiming success is quite difficult in this setting. So how do we set up all or none testing procedures in trials with this inferential goal? The solution is in fact quite straightforward, almost trivial, and I'm going to explain how it works on this slide. It only takes just one slide to define the appropriate class of procedures. Let us introduce the null hypothesis of no effect and the p-values based on appropriate statistical tests for the null hypothesis that are shown at the top of the slide. And alpha here will denote the overall type 1 error rate, which is the probability of incorrect conclusions. For example, a one-sided alpha of 0.025 could be used. And in the intersection union problem, all null hypotheses are rejected, which also means that their union or the global null hypothesis is rejected if all p-values are less than or equal to alpha. As a result, there is no multiplicity adjustment. All individual tests are carried out simply at the full alpha level, without any multiplicity penalties. And it is not because all or none procedures are efficient. In fact, as we will discuss that in a second, all or none procedures are actually quite conservative, if anything, and may lead to substantial power loss. What we see on the slide is the complete solution to the intersection union multiplicity problem. There is really nothing else to discuss, and this is in fact the last time we're going to see this problem in this course. On a related topic, I would like to point out that it is stressed in the FDA guidance that there have been suggestions that the statistical testing criteria for each co-primary endpoint could be relaxed. For example, testing could be performed at an alpha of 0.06 or 0.07. Here, of course, the authors of the guidance refer to two-sided alpha levels, and they're implying here that the two-sided alpha level of 0.05 could be potentially relaxed and increased to 0.06 or 0.07. However, it is pointed out in the guidance that relaxation of alpha is generally not acceptable, because doing so would undermine the assurance of an effect on each disease aspect considered essential to showing that the drug is effective in support of approval. So once again, as we said on slide 12, an inferential approach used in trials with co-primary endpoints is quite straightforward. Each endpoint must be tested at the predefined alpha, for example, one-sided alpha of 0.025, and this is the complete solution to the intersection union multiplicity problem. But before we close the door on all or none procedures, let us quickly illustrate procedures in this class. Let us assume that the Alzheimer's disease trial is complete. We have computed one-sided p-values for the two endpoints, and they're given, as you can see on the slide, by 0.0113, this is p1, for the cognition endpoint, and the p-value for the global clinical assessment endpoint is p2, which is equal to 0.0187. The p-values are plotted on the slide. They are represented by the orange dots. You can see here easily that both p-values are below the threshold that represents the one-sided alpha of 0.025. Each p-value is significant, and therefore the all or none procedure rejects both null hypotheses of no treatment effect. The trial is declared successful, and the sponsor can claim that the treatment significantly improves both cognition and global clinical assessment in patients with Alzheimer's disease. And another important point related to the analysis of co-primary endpoints, a well-known problem arising in clinical trials where all or none procedures are applied is that, as it is emphasized in section 3 of the FDA guidance, the use of two or more endpoints for which demonstration of an effect on each is needed to support regulatory approval, called co-primary endpoints, increases the type 2 error rate and decreases study power. In other words, with a large number of co-primary endpoints, the probability of success is reduced. This is a direct consequence of the fact that, as we said a few slides back, the parameter space for the global alternative hypothesis is quite small, and claiming success in this setting is quite difficult. And I would like to also add that it is further stated in the FDA guidance document that the loss of power may not be so severe when the endpoints are correlated, because in a trial with two positively correlated co-primary endpoints, there is an increased chance that the second endpoint will demonstrate a significant treatment effect if the first endpoint is significant, which leads to higher power. However, When considering use of co-primary endpoints in a study, it should be recognized that use of more than 2 co-primary endpoints can markedly reduce power in this trial. We're now done with the second set of inferential goals with intersection union problems, and we're ready to move on to the last inferential goal. In this last setting, individual analysis or individual objectives in a clinical trial with multiple objectives are components of an overall analysis. They are components of the same underlying variable, but they probably measure different aspects of that underlying variable. The global successful outcome in a trial is defined based on a combination of individual analyses, and the trial's outcome is positive if this overall effect is significant. I'm going to use example 3 from Module A, which is based on the fracture healing trial, because it serves as a good example of a global testing procedure. In this example, the underlying variable of interest was functional improvement in patients with osteoporosis, and changes in that variable were studied by evaluating the drug effect on 3 related endpoints. They were defined as time up and go test, 6-minute walking distance test, and pain score. In other settings, this global testing approach is applied when multiple endpoints are biologically related to each other, and they may be viewed as individual aspects of a single variable with a complex structure. This would be the case, for example, in clinical trials for the treatment of rheumatoid arthritis or other autoimmune diseases. We will not be discussing this global testing approach in this course, mainly because it is very uncommon to see it used in confirmatory clinical trials. In fact, I pointed out in Module A that when I introduced example 3, that this particular example is based on an unpublished phase 2 study. But I would like to say that much work has been done in this area, and there are several elegant statistical methods that could be used to build global testing procedures. If you would like to find out more about how to develop and apply global testing procedures, I would recommend that you read chapter 4 in our 2009 book, which is entitled Multiple Testing Problems in Pharmaceutical Statistics. Section B2, Error Rate Definitions for Multiple Testing Procedures. We're now done with the discussion of inferential goals in multiplicity problems, and in section B, in all other sections from this point on, we will focus on the at least one multiplicity problem, also known as intersection union multiplicity problem, and the most important feature to remember about this problem is that at least one testing induces error rate inflation. In section B3, we will talk about rules for selecting most appropriate multiple testing methods for a given clinical trial, and to choose an appropriate method, it is critical to know how to define correct and incorrect decisions in a problem, or, in other words, it is important to define the type 1 error rate. Well, we may ask, are there several definitions of the type 1 error rate? Yes, in fact, there are quite a few of them, and the preferred definition, the definition used in confirmatory clinical trials, the definition which is recommended in the regulatory guidance documents, is the family-wise error rate, also known as FWER. There are other definitions that you may have heard about, such as the generalized family-wise error rate, the false discovery rate, but they are not used in clinical trials. They make much more sense and they have found numerous applications in preclinical studies, in drug discovery, and here we'll focus on the family-wise error rate. Here's a quote from the EMA guidance document. This quote emphasizes the importance of controlling the type 1 error rate in confirmatory clinical trials using a multiplicity adjustment. It points out that no adjustment is necessary only if there are two treatment groups in a clinical trial, a single primary variable, a single null hypothesis, and no interim analysis. The guidance document, I believe, does a very good job of driving this message home. Addressing multiplicity issues in confirmatory trials is of paramount importance. Addressing multiplicity issues and, even more importantly, efficient solutions to multiplicity problems is a top consideration in clinical drug development, and the error rate definition recommended in the two regulatory guidance documents by the U.S. Food and Drug Administration and by the European Medicines Agency is the family-wise error rate, which takes us to the definition of this error rate. Let us now provide a formal definition of the family-wise error rate. We're going to consider a clinical trial with null hypotheses of no effect that are denoted by H1 through HM. And in confirmatory clinical trials, the family-wise error rate must be controlled in a strong sense at a pre-specified alpha level, for example, two-sided alpha of 0.05 or one-sided alpha of 0.025. This means that the probability of incorrectly rejecting at least one true null hypothesis is less than or equal to alpha, regardless of which and how many other null hypotheses are true. Well, it is quite a mouthful, and let us now try to digest this definition. The definition tells us that we need to preserve the type 1 error rate not only for the case when there is no evidence of a positive effect across the hypothesis of interest. Our goal here is to protect the error rate for all possible configurations, all possible combinations of true and false null hypotheses. Essentially, we're talking about the maximum type 1 error rate, the type 1 error rate for the worst case scenario. I think that an illustration will be very helpful at this point because it helps us answer this very important question. Why do we need such a potentially conservative definition? Why should we control the type 1 error rate for the worst case scenario? And the billion-dollar answer is, this definition enables us to make claims, specific claims about each individual outcome without inflating the error rate. And it is extremely important. For example, returning to example 4, based on the type 2 diabetes trial, let us define three null hypotheses, H1, H2, and H3. In this case, H1 will be the null hypothesis of no difference between the high dose, or dose 1 and placebo, and likewise H2 and H3 will be defined as the null hypothesis of no effect for the medium and low doses, or in other words, doses 1 and 2. And to control the family-wise error rate in a strong sense in this trial, we need to consider all possible combinations of true null hypotheses and show that the type 1 error rate is less than or equal to the pre-specified alpha level for any one of those combinations. For example, as you see here on the slide, suppose that the null hypotheses H1 and H2 are true and H3 is false, which means that the treatment effect is only present at the low dose, or dose 3. Then our definition of the error rate guarantees that the probability of rejecting, obviously incorrectly rejecting, those true null hypotheses cannot exceed alpha. So regardless of the true efficacy profile of this new treatment, and we don't know, by the way, and we will never know what this true efficacy profile looks like, regardless of that efficacy profile, we can draw general conclusions. For example, the new treatment is effective or it lacks efficacy, as well as we can make specific claims. For example, the low dose is effective, but the other two doses are not effective. The error rate definition takes the uncertainty about this true efficacy profile out of the equation, so to speak, and we don't need to worry about it anymore. Strong control of the family-wise error rate is a very powerful concept. And we can build a procedure that protects the family-wide error rate. We can use it anytime, anywhere, and we can trust the results. Let me now provide a quick summary. Strong control of the family-wise error rate enables clinical trial sponsors to make specific claims. The regulatory position is that procedures used in the primary analysis in all confirmatory clinical trials must guarantee strong family-wise error rate control. And given this, I'm sure you will not be surprised to learn that all multiple testing procedures we're going to discuss in this course actually control the family-wise error rate in the strong sense. The last section in this module is section B3, selection of multiple testing procedures. We have introduced so far the formal framework and defined inferential goals in clinical trials with multiple objectives. We have talked about key concepts behind at least one procedures. We've talked about the error rate control and most commonly used definition of type 1 error rate control in a clinical setting. These were theoretical considerations. This was the foundation of the multiple testing theory. And now the time has come for us to move on to more practical considerations. In this section, section B3, we will go over the process, the ideas behind selection of multiple testing procedures. This will be a general set of guidelines that will help us answer the question that we face every day. We encounter a clinical trial with multiple objectives. The question is, we know we need to control the family-wise error rate in the strong sense. It's a regulatory requirement. But which multiple testing procedure should we use? There are lots of choices. Which is the best choice? To answer these questions, we will go through the following steps. Step 1, we will define hypothesis testing problem, which means that we will define the individual null hypothesis that will be tested. Step 2, we will define the relationships among the null hypotheses of interest. These are known as logical relationships or logical restrictions. For example, the null hypothesis may be naturally ordered. We need to know this. We will need to ask this question. This will help us narrow down the choices. And secondly, there are also distributional assumptions, distributional relationships. For example, the question to ask would be, what is the joint distribution of the hypothesis test statistics? Is it known to us? Or is there any uncertainty about it? Can we assume that it is known and then we can utilize this knowledge to build a multiple testing procedure? The next step is step 3. Using the information from steps 1 and 2, we will define one or more candidate multiple testing procedures that are consistent with the logical restrictions as well as the distributional relationships. And finally, step 4, the last step here, is to select the most powerful procedure from the set of candidate procedures. Well, to begin illustrating the decision-making process we have just defined, let us now apply it to several multiplicity problems. We're going to use the same notation that we've used before. H1 through Hm will denote the null hypothesis of interest, and the family-wise error rate will be denoted by alpha. For example, we will assume that the one-sided alpha is equal to 0.025. We will assume for simplicity that the null hypotheses, the M null hypotheses of interest, are equally important or they're equally weighted. It will be in fact quite easy to extend the theory that we're going to discuss to the case of unequally weighted hypotheses. We're also going to introduce the hypothesis test statistics used for testing the hypothesis of interest. Those will be derived using appropriate statistical tests for each individual hypothesis. They will be denoted as we see on the slide by T1 through Tm. And in addition, when discussing more powerful multiple testing procedures that are known as stepwise procedures, we will make use of ordered test statistics. These are defined using the following parenthetical notation. The ordered test statistics are arranged from the largest one or the most significant one to the smallest or the least significant statistic. And similarly, we can define p-values computed from the null distributions of these test statistics. When stepwise procedures will be introduced, we will differentiate between the original p-values as well as ordered p-values. The ordered p-values will be arranged from the smallest one to the largest one, as you see at the bottom of the slide. Now, as we continue to step 2, the following two classification schemes play an important role in the world of multiple comparisons, and they will help us understand two different sources of information that influence the choice of multiple testing procedures in each individual clinical trial. The first classification scheme is based on available clinical information, which defines clinically relevant logical relationships or restrictions among the null hypotheses of interest. Based on this information, as I will explain in a few minutes, we can introduce two different classes of multiple testing procedures, known as single-step and stepwise procedures. And secondly, there's a classification scheme which is based on available statistical information. This classification scheme is driven by distributional relationships, which means the available information on the joint distribution of the hypothesis test statistics. And depending on how much information we have, we can consider nonparametric, semiparametric, and fully parametric procedures. We're going to define those classes of multiple testing procedures later in this module, and we'll provide multiple examples later in this course. Let us first discuss the classification scheme that relies on the available information on clinically meaningful logical relationships or logical restrictions among the null hypotheses in our multiplicity problem. In Module C, we will introduce basic procedures such as the Bond-Ferroni procedure that utilize the single-step testing approach. Within this approach, the null hypotheses are tested simultaneously, or in other words, they're tested in a single step. Clinically meaningful relationships among the null hypotheses are not actually taken into account, and this is of course a limitation which ultimately leads to lower power. And by contrast, when the stepwise testing approach is applied, the null hypotheses are ordered using clinically relevant criteria such as clinical importance or using significance of the test statistics or p-values. And this is where the ordered test statistics or ordered p-values that we introduced a few minutes ago may be used. Within this class of multiple testing procedures, we will draw a line between procedures with a pre-specified testing sequence and a data-driven testing sequence. We will take a closer look at those two classes of stepwise procedures in a minute. But first, we will quickly discuss basic non-stepwise procedures or single-step procedures. The decision to reject any null hypothesis, in this case, does not depend on decision rules for the other null hypothesis. There is really no testing sequence. Each null hypothesis is rejected independently of the other null hypothesis. In this diagram, each hypothesis is, in this multiplicity problem, is represented by an orange box, and with any single-step procedure, you can test the hypotheses in any order. They are not arranged in any meaningful sequence. And examples of single-step procedures are shown at the bottom of the slide. The Bond-Ferroni and Dunning procedures that we're going to discuss later in this course serve as very good examples of the basic single-step testing approach. We will use this illustration to discuss the role of logical relationships among null hypotheses in a clinical trial. It is important to know whether or not there is a natural way to order the null hypothesis in a multiplicity problem. Suppose first that we can select a pre-specified testing sequence, or in other words, a pre-specified hypothesis ordering, which means that the null hypotheses H1 through Hm are arranged, they are ordered according to their clinical importance or probability of success. In this case, we say pre-specified to emphasize the fact that we have enough prior information to choose a testing sequence before we unblind the data in a clinical trial, before we actually see, before we compute the hypothesis test statistics. And once the testing sequence is fixed, it cannot be modified. It is set in stone, and we build a chain, and we cannot break it to rearrange the links. Example 4, based on the diabetes trial, serves as a good example. In this case, for example, let us suppose that we're planning a phase 3 trial and we have the results from the recently conducted phase 2 trial. There is strong evidence of a positive dose-response relationship in this study. And given this information, knowing that the effect size is likely to be maximized at the high dose, we place the associated null hypothesis H1 at the beginning of the sequence. This approach helps us improve the probability of success, the probability of detecting a significant effect. The next null hypothesis in the sequence could be H2, because the medium dose is expected to be less effective than the high dose, but at the same time more effective compared to the low dose. Then H3 will be the last hypothesis in the sequence. So, to reiterate, with a pre-specified testing sequence, the null hypotheses in a clinical trial are ordered and tested sequentially. As you can see in this diagram, a stepwise procedure with a fixed testing sequence begins with H1 and then it goes down the sequence. In Module D, we will define 3 stepwise multiple testing procedures of this kind, or I should actually say that some of them are really classes of multiple testing procedures. This includes the fixed sequence procedure, a class of fallback procedures, and a class of chain procedures. Now, we will take a look at the same multiplicity problem, but from a different perspective. Let us assume that there is not enough information to order the null hypothesis of interest in this clinical trial. In this case, we will have to rely on a data-driven testing sequence when we build a multiple testing procedure. For example, if we take another look at example 4, and I intentionally use the same example so that we will have the same multiplicity problem as before, but the multiple testing approach may be different because the logical relationships among the hypotheses are different. So using example 4 as an example, it is difficult to assume a positive dose-response relationship. In other words, it is risky to assume that the largest effect size will be observed at the highest dose, or dose 1, and then the effect size will decrease as we step down to the medium dose and then to the last dose. With this prior information, it makes more sense to let the 3 null hypotheses H1, H2, and H3 be tested in the order determined by the significance of the hypothesis test statistics, as it will be shown on the next slide. With a data-driven testing sequence, the null hypotheses, as we said a second ago, in a general multiplicity problem are tested sequentially, but the hypothesis ordering or testing sequence are determined only after we unblind the data and compute the hypothesis test statistics. This is why we call it a data-driven hypothesis ordering. The null hypotheses are tested in the order determined by the significance of the hypothesis test statistics. For example, it may turn out that the data-driven sequence begins with H2, followed by Hm, which is the last hypothesis, and H1 may end up being at the very end of the sequence. We will introduce several stepwise multiple testing procedures based on this type of logical relationships among the null hypotheses. This includes the Holm, the Hummel, the Hogberg procedures in Module C, and then in Module E we will talk about the step-down Dunning procedure. Another important critical consideration in the process of selecting candidate multiple testing procedures is distributional relationships or assumptions on the joint distribution of the hypothesis test statistics in a multiplicity problem. We will begin with this diagram and then we'll go over each individual type of distributional assumptions. Very quickly, depending on the distributional assumptions, there are three classes of multiple testing procedures. There are nonparametric procedures, there are semiparametric, and there are fully parametric procedures. Beginning with the nonparametric procedures on the left, they make no assumptions about the joint distribution of hypothesis test statistics. We can think about them as distribution-free procedures. In a problem with two tests that are represented here by two orange boxes, test 1 and test 2, we do not assume any connection between the two test statistics. There is just empty space between the boxes. Each test is simply based on its own univariate p-value. Now I'm going to jump all the way to the right to the class of parametric procedures that make specific or explicit distributional assumptions. They make full use of the underlying joint distribution of hypothesis test statistics. And to help you remember this, we will connect the two tests, the two orange boxes here, with a solid line. The individual tests are connected within the parametric testing framework because the correlation structure, the joint distribution, is fully known and fully taken into account when a parametric procedure is set up. And the last class of multiple testing procedures is shown in the middle. It is the class of semiparametric procedures. Unlike the parametric approach to constructing multiple testing procedures, the semiparametric approach does not assume that the joint distribution of test statistics is fully specified. For example, we may assume in a certain clinical trial that the test statistics follow a multivariate normal distribution, but the correlation coefficients are unknown. In this sense, the semiparametric approach is somewhere in between the parametric approach on the right and the nonparametric approach to constructing multiple testing procedures, and it is shown on the left. Since the joint distribution of test statistics is only partially specified, we will connect the two tests in this diagram using a dotted line. In other words, within the semiparametric framework, there is a connection among the test statistics, but it's a kind of a dotted connection. Some parameters of the joint distribution may be unknown. Here is a more detailed summary of the three classes of multiple testing procedures, beginning with the class of nonparametric procedures. Nonparametric procedures, as we said, rely on basic probability inequalities. For example, the bond-front inequality to control the family-wise error rate, and this is why they do not need to make any assumptions about the joint distribution of hypothesis test statistics. Examples include the bond-front, Holm, fixed sequence, fallback, chain procedures. They're all non-parametric procedures. I'm sure you're going to recognize quite a few procedures on this list, and this is because non-parametric procedures are very popular. They enjoy much popularity in clinical trials because they're intuitive, they're easy to apply, and they're easy to explain to non-statisticians. But, of course, the question to ask for us now is, what are the downsides? Non-parametric procedures tend to perform poorly with a large number of null hypotheses or strongly correlated test statistics because they do not connect the dots, so to speak. They ignore the fact that there is certain joint distribution behind the individual test statistics, that the hypothesis test statistics, for example, are correlated. Since this class of procedures tends to ignore the underlying correlation structure, they are less powerful than multiple testing procedures that make certain distributional assumptions. For example, semiparametric or parametric procedures that I'm going to introduce on the next two slides. Semiparametric procedures serve as a more powerful alternative to basic nonparametric procedures. They're also based on univariate p-values, but unlike their nonparametric counterparts, they impose some distributional assumptions. For example, what do I mean by some? For example, a multivariate normal distribution for the test statistics may be assumed, but the correlation coefficients may be unknown. The Hochberg and Hummel procedures are the most widely used, most popular semiparametric procedures in clinical trial applications. These procedures are more powerful than nonparametric procedures such as the Holm procedure. Moving on to parametric procedures, this is a class of exact multiple testing procedures that make specific distributional assumptions. For example, they may assume that the hypothesis test statistics in a multiplicity problem follow a multivariate normal distribution or a multivariate t-distribution. Parametric procedures rely on multivariate p-values that are computed from those pre-specified joint distributions. And when we say parametric procedures, we typically immediately think of the Dunnett procedure developed by Charles Dunnett back in 1955, but there are also multiple related parametric procedures. There's actually a family of Dunnett procedures that includes the basic or single-step Dunnett procedure. There's also a step-down Dunnett. There's also a step-up Dunnett procedure. They will be introduced in Module E. And since parametric procedures make full use of the joint distribution of hypothesis test statistics, they are more powerful than non-parametric procedures. But what we need to keep in mind is that parametric procedures, unlike their semiparametric counterparts, they're very sensitive to the underlying distributional assumptions. Parametric procedures are recommended whenever the underlying parametric assumptions can be justified. For example, when the joint distribution of test statistics is fully known to us prior to the start of a trial. But these procedures will break down, and as a result, the type 1 error rate may not be controlled if these parametric assumptions are somehow violated. So we need to be careful. I would like to very quickly mention that in addition to non-parametric, semiparametric, and fully parametric procedures defined earlier, it will be worth mentioning the class of the so-called resampling-based procedures. These procedures do not make explicit distributional assumptions, and what they rely on certain approximations to the true joint distribution of the hypothesis test statistics. And this is accomplished by using resampling-based methods, for example, the bootstrap or permutation methods. Resampling-based procedures essentially rely on approximation when evaluating the null distribution of the test statistics, and from a theoretical perspective, they can guarantee type 1 error rate control only asymptotically when the sample size in a trial goes to infinity. When it comes to confirmatory trials, the FDA guidance on multiple endpoints makes it clear that resampling methods are not recommended as primary analysis methods for adequate and well-controlled trials in drug development. Section 4 of this guidance document states that there is at present little experience with these methods in drug development, clinical trials. Because of this, resampling methods are not recommended as primary analysis methods for adequate and well-controlled trials in drug development. It may, however, be useful and instructive to compare the results of resampling methods with those obtained using conventional methods in order to gain experience with and understanding of resampling methods, their properties, advantages, and limitations. But at the same time, I would like to add that resampling-based procedures have actually found applications in clinical drug development. It's just not in confirmatory trials. For example, they've been successfully used in the so-called thorough QT studies that are conducted for virtually all compounds to assess certain cardiovascular risks, and resampling-based procedures have been applied to construct procedures that control the type 1 error rate in the assay sensitivity analysis with a positive control when the positive control is compared to placebo. And this is just a quick example for your information. At this point, it will be instructive to review issues that frequently arise in the context of distributional relationships in clinical trials. These are the kinds of considerations that play a key role in the process of selecting most appropriate multiple testing procedures for a particular problem arising in a clinical trial. Let us consider the multiplicity problem in example 1 based on the prostate cancer trial, where multiplicity is induced by the analysis of two primary endpoints based on radiographic progression-free survival and secondly on overall survival. The question we would like to ask here is, can we use a nonparametric, semiparametric, or parametric procedure in this trial to protect the type 1 error rate? Before we answer the question posed on slide 41, let us step back a bit and think about the distributional information available in the prostate cancer trial. The two endpoints in this trial, progression-free survival and overall survival, may be positively correlated, and if they are indeed positively correlated, it would be beneficial to take advantage of this information, because with this information we could have constructed a parametric multiple testing procedure that would have led to a powerful, efficient multiplicity adjustment. A very important consideration here is that correlation information of this kind can be taken into account only if it is known at the trial's design stage. I've already alluded to this fact when I introduced the class of parametric procedures. I said that the joint distribution of test statistics must be known prior to the start of a clinical trial. Of course, the correlation between overall survival and progression-free survival can be estimated, but use of sample correlations in multiple testing procedures may result in error rate inflation. This is a very important point for us to bear in mind. Even though the FDA multiplicity guidance provides a detailed coverage of multiplicity issues arising in the context of clinical trials with multiple endpoints, it does not touch upon this important issue, a bit surprisingly. It never mentions an option to set up a multiplicity adjustment that uses sample correlation coefficients among the endpoints. It does not go over the pros and cons of this approach. However, this is an issue that has attracted attention in the clinical trial literature. For example, there is a relevant quote from a paper written by 3 FDA statisticians, and it is emphasized in this paper that the use of correlation for alpha allocation may be challenged when the trial is not prospectively planned with a sample size to detect a pre-specified treatment effect in the subset, in which case the sample size fraction is unknown at the trial design stage and is not determined until the end of the study. This is a recommendation that applies to clinical trials with multiple patient populations, but the same type of argument could be used in clinical trials with multiple endpoints. This recommendation makes good sense, and it is consistent with the considerations that we discussed a minute ago on slide 42. As a quick illustration, let us take a closer look at example 1 with 2 endpoints Let us construct a parametric procedure for addressing the multiplicity problem in this clinical trial with the PFS and OS endpoints. We're going to assume that the two test statistics follow a bivariate normal distribution. I would like to point out that not the endpoints themselves, but the test statistics, the log rank test statistics that are used to assess the evidence of a beneficial effect. Those test statistics follow a bivariate normal distribution, and we can estimate the correlation between the endpoints. Let us say that the sample correlation rho is 0.8, and we will use this estimate to compute the critical value for the parametric procedure. Using this critical value, we will compute the family-wise error rate of the resulting multiple testing procedure, and it will be a function of the true correlation coefficient, which is unknown to us. We've estimated it, and the sample correlation coefficient is 0.8, but the true correlation could vary, could range from 0 to 1. And this plot provides a graphical summary of the key characteristics of the resulting parametric multiple testing procedure. We see here that when the true correlation equals the sample correlation, the parametric procedure does a perfect job of preserving the error rate. The one-sided error rate is exactly 0.025. If the sample correlation overestimates the true correlation, the parametric procedure becomes conservative in the sense that the actual error rate is going to be below 0.025. So it becomes somewhat inefficient, but at least it controls the family-wise error rate. What is worse is that when the sample correlation underestimates the true correlation, things get out of control. The solid curve, as we see here on the slide, crosses the 0.025 threshold. The multiplicity adjustment does not work anymore. And this is why it is not safe to use sample correlations when building multiple testing procedures. We're now ready to return to the question, can we use a nonparametric, semiparametric, or parametric procedure in this trial to protect the type 1 error rate in the prostate cancer trial? The answer depends on what we know about the two test statistics associated with the primary endpoints in this trial. It will be ideal, as we have said, to apply a parametric procedure because it is the most efficient approach to performing multiplicity adjustments, and it results in highest power. But the general parametric approach may be used only if the correlation between the endpoints is known at the design stage, which means it's known prior to the beginning of the study. Since this is not the case, we don't know what the true correlation coefficient is, we cannot apply a parametric procedure, and we will need to consider alternative approaches. A semi-parametric procedure may be used in this trial if the correlation between the endpoints is known to be non-negative. But it is not generally clear how to justify this assumption. It is theoretically possible that the two endpoints may not be positively correlated. For this reason, it appears to be most appropriate in the context of this particular clinical trial to resort to a non-parametric procedure. After all, by definition, a non-parametric procedure can be applied to any multiplicity problem. The correlation between the endpoints may be unknown, but this will not be a problem within this class of multiple testing procedures. We have a few slides left in this module, and these slides deal with steps 3 and 4 of the general set of guidelines to facilitate the selection of most appropriate multiplicity adjustment for a given multiplicity problem. We will just take a quick look at the two steps because we will return to them in other modules later in this course. In step 3, I would like to remind you we are expected to identify a candidate set of procedures consistent with the clinical and statistical requirements defined in step 2. On the next 3 slides, I will very quickly give examples of multiple testing procedures based on the two classification schemes I introduced earlier in this module. Let us begin with a class of single-step procedures. When it comes to nonparametric procedures, the Bonferroni is probably the best example, and the Bonferroni procedure will be discussed in Module C. There are two examples of semiparametric single-step procedures. It is SIMES and SHIDAC. The SIMES test will be discussed in Module C. The SHIDAC test will not be discussed. It is very uncommon to see it applied in confirmatory clinical trials. And when it comes to parametric procedures, the standard, the basic Dunning procedure, relies on a single-step testing approach, and it will be discussed in Module E. This slide takes a closer look at stepwise procedures that rely on a pre-specified hypothesis ordering. As before, we can talk about examples of nonparametric, semiparametric, and fully parametric procedures. Beginning with the first class, examples include the fixed sequence, fallback, and a class of chain procedures that will be introduced in Module D. There are really no examples of semiparametric procedures that rely on a pre-specified hypothesis ordering. And the last class, the class of parametric procedures, is represented by the so-called parametric fallback and parametric chain procedures. They will not be discussed. They will be beyond the scope of this online training course. And finally, very quickly, a class of stepwise procedures that rely on a data-driven testing sequence. Examples include a non-parametric procedure such as the Holm that will be again presented in Module C. There are two semi-parametric procedures, the Hogberg and the Hommel, that will be discussed in Module C. And in Module E, we're going to introduce two parametric procedures that rely on a data-driven testing sequence, and those are known as the step-down Dunning and step-up Dunning procedures. I would like to point out that this classification scheme based on logical restrictions is not perfect, just like any other classification scheme. To give a quick example, many procedures from the class of chain procedures actually rely on a data-driven testing sequence. This is due to the fact that the class of chain procedures is quite broad, and indeed it includes, for example, the Holm procedure, which is shown here under stepwise procedures with a data-driven hypothesis ordering. Despite these imperfections, I believe that this classification scheme facilitates the general decision-making process when a clinical trial sponsor faces the problem of selecting most appropriate multiple testing procedures for a particular multiplicity problem. We're almost done with Module B. This is the last slide in the module. And in step 4, we focus on the very challenging problem of assessing the available multiplicity adjustment options. For example, reviewing the candidate procedures identified in step 3, selecting an optimal, most efficient multiple testing procedure for a given problem in a given clinical trial. Since it's a very general and open-ended task, I will present here only a set of general recommendations based on my general experience with dozens and dozens of multiplicity problems in phase 3 confirmatory clinical trials. First of all, based on the first classification scheme that utilizes the available information on logical restrictions among the null hypotheses, we could consider a multiplicity adjustment based on a single-step procedure or a stepwise procedure. Here, finding more efficient procedures is actually quite easy. We know that stepwise procedures are more powerful than single-step procedures, and in addition, it is generally true that stepwise procedures with a data-driven testing sequence are flexible and therefore they are more robust than stepwise procedures with a fixed or predefined testing sequence. And the second classification scheme that we defined in this section is based on assessing the amount of distributional information For example, how much we know about the joint distribution of the hypothesis test statistics. We have defined nonparametric, semiparametric, and parametric approaches to building multiple testing procedures. And again, in general, semiparametric procedures tend to perform the best because they strike a balance between nonparametric and parametric approaches in the sense that semiparametric procedures are more powerful than their nonparametric counterparts, and at the same time, semiparametric procedures are more robust than parametric procedures.